Okay, so in this topic, we're going to look at the synthesis of a specific polypeptide. But the thing here, if we do know that the gene does not synthesize a polypeptide directly, so means that we need to have the bridge or intermediate between the DNA and polypeptide is our RNA. So in this case, you can see that involve three kinds of RNA. Correct or not? We involve the mRNA, messenger RNA. We involve tRNA, the transfer RNA. Then another type is ribosome, rRNA. So total involve three types of RNA in the process to from DNA to become polypeptides. And the process involved here, they are transcriptions and translations. Okay, so two processes involve transcriptions, translations. Okay, this is the part, right? Now we start first. So we have the DNA triple codes, mRNA codon, and tRNA anticodons. So the code here always three letters. So we call it triple codes for DNA triple codes. Now, highlight here, special in this part. If you want to read the DNA triplet, we always read it in 3' to 5' direction, guys. So student always confused. So you say that 5' to 3', always 5' to 3'. Why suddenly 3' to 5'? Always remember, if you want to elongate in the process of elongations of the strand, make it longer, like DNA replications, mRNA synthesis. When you want to elongate something, make it longer, always remember it must be 5' to 3'. Are you clear? You want to add something, see that. But when I want to read, I can read it from A to Z. I can read it from Z to A. When I want to read it, are you clear? But for DNA triplets, we always read it in 3' to 5' directions. Always remember, DNA triplet is always read how we read it. It must be in 3' to 5' direction. It's very important. If you read the opposite way, then you will get a wrong polypeptide. For example, AAT and TAA definitely be code for different amino acids. Are you clear? Okay. So the question is why? Why we read 3' to 5', not 5' to 3'? Because mRNA, ribosome reads mRNA or translates the mRNA 5' to 3'. And mRNA is anti-parallel to my DNA. So that's why if you read your mRNA 5' to 3', therefore you read the DNA triplet codes from 3' to 5' because anti-parallel properties. Are you clear? Okay. So it depends on the question. It depends on the question. For example, you look at this activity 10.14. This is a template strand. Now, if this is a template strand and is given 5' and 3' here, always remember how we read it. We read it always from 3' to 5'. Are you clear? Now, two kind of questions. One question will give you 5' to 3'. Then you note, always read from the 3' to 5'. It depends where I put the 3' are. Are you clear? Okay. Now, if question did tell us 5' or 3', for example, try to example, blank out the 5', blank the 3', then conventionally always left, right. Clear or not? Okay, now guys, it depends how question is designed. If the question tells us 5', 3', then 3' to 5'. Are you clear? Okay, because this DNA triplets. If the question does not give us the 3', 5', it straight away start CTC, CTC, AGG, and so on. Then we always follow conventional left to right. Are you clear? Okay. depends on the type of question. If the question tells us where is a 3' and 5', always read 3' to 5'. Very clear? If the question didn't tell us where is a 3', where is a 5', then easier left to right. Okay? So now this is DNA triplets. What I want you guys to do now, very, very simple. Based on this, okay, so figure out the sequence of mRNA and the sequence of the polypeptides. Oops, sorry. My mistake is we cannot be sequence of polypeptide, sequence of the amino acid. Okay. Okay, so this is the answer, sequence of the mRNA and also the sequence of amino acids. Always remember we read it from 3' to 5' ends when you read it. Okay, correct? Any mistake? No, right? Am I doing any mistake? Hopefully no. Okay. Now you have the mRNA codon table. Now what's this mean? Yes. Oh, wait. 1, 2, 3, 4. This one. TGA. Oh, TG. So isoleucine. TG. Oh, threonine. Sorry. THR. Threonines. Okay, sorry guys. Okay, my mistake. Any questions so far, guys? Okay. Now, it's easy when the question gives you the mRNA, you find out from the mRNA table. Can I see that? But questions sometimes they're not so generous to you guys. What they do, they give you DNA, but give you mRNA table. So if you look this type of question given, then you have to use here. For example, GUG 5', you have to read mRNA from 5' to 3'. So GUG. So GUG valent. It's still the same. Are you clear? It will be the same. Are you clear? So that's why, guys, you have to be extra careful whether it's DNA triplets or mRNA codon. So how we know, guys? How we know which one question give you? Give you DNA triplets or give mRNA codon? One thing, very simple. Check whether got T and U or not. If the sequence contains T, must be DNA. If the sequence give you contain U, it must be mRNA. If the sequence contains T and U, you just write down wrong question. Are you clear? It cannot be, right? How can your mRNA got T? How can your DNA got U? So if you look at the sequence, they give you got T and U, just write down. Stupid error. Okay, shame on you. Okay, I get 0 marks, then none of my business, huh? Okay, so but basically it won't be like this unless typo. I don't think T and U is side by side. Okay, so but basically you have to understand that. What I mean here is how you know whether it's the DNA sequence given or mRNA sequence given. If the DNA sequence given, how we know? Check for T. Are you clear? Okay, question won't give you CCGG, no T at all. It cannot be. Are you clear? Okay, so how you know it's mRNA? We check for the U. If DNA, how we read it? DNA we read from 3' to 5'. If mRNA, opposite, 5' to 3'. Okay. So these anticodons, no need to worry about this because it won't form a sequence because only 3 codes. Are you clear? We use entire tRNA. We only use the last part, the 3 code only. So in this case, a 3-digit code or 3-letter code only. So it won't form a sequence, long sequence. So don't worry about tRNA at all. Are you clear? Okay. So the codes on the DNA molecules used to determine the sequence of the amino acid, and we said that in the polypeptide. So that's why we always think about it. How can our nucleus control the cellular activity? Right. control cellular activity. How they know which to do, what to do. So basically, always remember, now we combine two topics. First, all biochemical reactions in our body, they have very high activation energy enzyme. Are you clear? So therefore, we need enzyme to lower down the activation energy. So how we do this? Then we need to have our nucleus. Inside the nucleus contains the DNA. Inside the DNA, segments of DNA contain the gene. So this gene can be transcribed into mRNA, and this mRNA now enter into the cytoplasm, bind with the ribosome, translation process takes place. Based on the mRNA codon, we can translate mRNA codons into a sequence of the amino acid, forming a polypeptide. So this polypeptide will fold into a precise three-dimensional shape, forming the enzyme. So with the present enzyme, biochemical reactions can take place. So cellular response takes place. Are you clear? So this is how our nucleus actually control the cellular activities. Okay, so with this, I have done for this topic. But before we end, I just want to show you guys. Again, I say that you do not need to memorize the table. But one interesting part here, I want you guys to write down. Highlight few items here. First, methionine, which is TAC, the code TAC for methionine. You realize that methionine is the only, I think you have two. One is the methionines, another one is the tryptophan, TRP and methionine. The rest, at least they have two codes. You look at this. If leucine, we have 6 of them. Can you see that? If you look at isoleucine, we have 3 of them. If you look at proline, we have 4 of them. If you look at the glutamines, we have 2 of them. Can you see that? Are you clear? So it means that some amino acids they have more than 1 code, and this is very, very important because you try to imagine if mutation takes place. Let's say, for example, mutation takes place, change the GTT to GTC. It still could back the same amino acid. Are you clear? So that's advantage of having multiple code for one amino acid. Now, why I point out methionine? Methionine also, TAC also represent the start triplet. What does this mean? It's the start triplet. It's the starting point of the translation. So generally, all protein or all polypeptide start with methionine. But after that, we have the modification to remove the methionine if we do not need it. Okay, remember guys, all polypeptide always start with methionine first. Start with methionine. Then methionine, after that, threonine or whatever, blah, 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 blah. Finish. If we need it, we keep it. If we don't want to keep it, or we don't want, we remove it. Are you clear? So removal of the methionines always takes place inside the brown sugar, which we didn't learn about this in your first chapter. Because your first chapter, I tell you removing methionine, everyone will look at it, what is methionine? Are you clear? Okay, so here now we know that methionine always the first one, but we remove it. Okay, now how we know where to stop? So highlight, we have these 3 codes here called stop. We don't have amino acid term as stop, yeah? Okay, so this amino acid term as stop doesn't mean that we have the amino acid called stop. So basically, when the ribosome reads, okay, ribosome cannot read about cover ATT or okay, for example, UAA. UAA is a stop codon. Now, if we read ATT, so UAA, UAA is stop codon. So when the UAA, we don't have the tRNA to bring the amino acid. So therefore, the end of the translation process. Okay, so we have special these 4 things that we need to know. Okay, so with this, I've done for today's topics.